and welcome to my blog. For today's blog, I will discuss about cell cycle and cell division.
Let's go on to our discussion, the cell cycle. The cell cycle is an ordered series of events involving cell growth and cell division that produces two new daughter cells. Cells on the path of cell division proceed through a series of precisely timed and carefully regulated stages of growth, DNA replication, and the division that produces two identical cells. The cell cycle has two major phases, the enter phase and the mitotic phase. Enter phase. During the enter phase, the cell undergoes normal growth processes while also preparing for cell division. In order for a cell to move from enter phase into a mitotic phase, many internal and external conditions must be met. There are three stages of enter phase. These are called G1, S, and G2. G1 phase, or first gap. The first stage of enter phase is called the G1 phase or first gap because, from a microscopic aspect, little change is visible. However, during the G1 stage, the cell is quite active at the biochemical level. The cell is accumulating the building blocks of chromosomal DNA and the associated proteins, as well as accumulating sufficient energy reserves to complete the task of replicating each chromosome in the nucleus. S phase, or synthesis of DNA. Throughout enter phase, nuclear DNA remains in a semi-condensed chromatin configuration. In the S phase, DNA replication can produce through the mechanism that result in the formation of identical pairs of DNA molecule sister chromatids that are firmly attached to the centromeric region. The centrosome is duplicated during the S phase. The two centrosome will give rise to the meiotic spindle, the apparatus that orchestrates the movement of chromosome during mitosis. At the center of each animal cell, the centrosome of animal cell are associated with a pair of rod-like objects, the centriole, which are at right angles to each other. Centrioles help organize cell division. Centrioles are not present in centrosome of other eukaryotic species, such as plants and most fungi.
Phase or second gap. In the G2 phase, the cell replenishes its energy store and synthesizes proteins necessary for chromosome manipulation. Some cell organelles are duplicated and the cytoskeleton is dismantled to provide resources for the mitotic phase. There may be additional cell growth during G2. The final preparations for the mitotic phase must be completed before the cell is able to enter the first stage of mitosis. The mitotic phase. The mitotic phase is a multi-step process during which the duplicated chromosomes are aligned, separate, and move into two new identical daughter cells. The first portion of mitotic phase is called karyokinesis or nuclear division. The second portion of the mitotic phase, called cytokinesis, is the physical separation of the cytoplasmic components in the two daughter cells. Karyokinesis. Karyokinesis, also known as mitosis, is divided into a series of phases: prophase, prometaphase, metaphase, anaphase, and telophase that result in the division of cell nucleus. Prophase. During the prophase or first phase, the nuclear envelopes start to dissociate into small vesicles, and the membranous organelles fragment and disperse toward the periphery of the cell. The nucleolus disappears. The centrosome begins to move to the opposite poles of the cell. Micro microtubules that will form from the mitotic spindle extend between the centrosomes, pushing them farther apart as the microtubule fibers lighten. The sister chromatids begin to coil more tightly with the aid of condensant proteins and become visible under the light of microscope. Prometapase. During prometapase, the first change phase, many processes that were begun into prophase continue to advance. The remnants of the nuclear envelope fragment, the mitotic spindles continue to develop as more microtubules assemble and stretch across the length of the former nuclear area. Chromosomes become more condensed and discrete. Each sister chromatid develops a protein structure called the kinetochore. Metaphase. During metaphase, or the change phase, all the chromosomes are aligned in a plane called the metaphase plate, or the equatorial plane, midway between the two poles of the cell. The sister chromatid are still tightly attached to each other by cohesin proteins. At this time, the chromosomes are maximally condensed. Anaphase. During anaphase, or upward phase, the cohesin protein degrade and the sister chromatids separate at the centromere. Each chromatid, now called a chromosome, is pulled rapidly toward a centrosome to which its microtubule is attached. The cell becomes visible elongate as the polar microtubules slide against each other at the metaphase plate where they overlap. Telophase. During telophase or distance phase, the chromosome reach the opposite poles and begin to decondense, relaxing into chromatin configuration. The mitotic spindles are depolymerized into tubulin monomers that will be used to assemble cytoskeletal components for each daughter cell. Nuclear envelopes form around the chromosomes and nucleosomes appear within the nuclear area. Kinesis. Cytokinesis or cell motion is the second main stage of the mitotic phase during which cell division is completed via the physical separation of the cytoplasmic components into two daughter cells. Division is not complete until the cell components have been apportioned and completely separated into the two daughter cells. Although the stages of mitosis are similar for most eukaryotes, the process of cytokinesis is quite different for eukaryotes that have cell walls, such as plant cells. G0 phase. Not all cells adhere to the classic cell cycle pattern, in which a newly formed daughter cell immediately enters the preparatory phases of interphase, closely followed by the mitotic phase. Cells in G0 phase are not actively preparing to divide. The cell is an acquiescent stage that occurs when cells exit the cell cycle. Some cells enter G0 temporarily, unit an external signal triggers the onset of G1. Other cells that never or rarely divide, such as mature cardiac muscle and nerve cell, remain in G0 permanently. Cell division. Cell division is the process cells go through to divide. There are two types of cell division, which is prokaryotic cell division and eukaryotic cell division. Prokaryotic cell division. Prokaryotes replicate through a type of cell division known as binary fission. Prokaryotes are simple organisms with only one membrane and no division internally. Thus, when a prokaryote divides, it simply replicates the DNA and split in half. Eukaryotic cell division. Eukaryotic organisms have membrane-bound organelles and DNA that exist on chromosome, which makes cell division harder. Eukaryotes must replicate their DNA, organelles, and cell mechanism before dividing. Many of cells divide using process that is essentially binary fission. Cell division stages. Mitosis stage. Mitosis starts with prophase in which the chromosome is condensed. The cell proceeds to metaphase where the chromosomes are aligned on the metaphase plate. Then the chromosomes are separated in anaphase and the cell cytoplasm is pinched apart during telophase. Cytokinesis is the final process that breaks the cell membrane. Meiosis stage. The stage of meiosis are similar to mitosis, but the chromosomes act differently. Meiosis has two phases, which include two separate cell division without the DNA replicating between them. Meiosis first and meiosis second have the same four stages as mitosis: prophase, metaphase, anaphase, and telophase. In prophase 1, the chromosomes are condensed. In metaphase 1, the chromosomes line up across from their homologous pairs when they are separated in anaphase 1 and telophase 1. There is only one form of each gene in cell, known as reduction division. Meiosis 2 proceeds in the same manner of mitosis, which sister chromatids dividing in the metaphase plate. By telophase 2, there are full cells, each with half of the alleles as the parent cell and only a single copy of the genome. The cells can now become gametes and fuse together to create new organism. Meiosis 2 proceeds in the same manner as mitosis, which sister chromatids dividing on the metaphase plate. By telophase 2, there are four cells, each with half of the alleles as the parent cell and only a single copy of the genome. The cells can now become gametes and fuse together to create